applicable across the globe depending upon the ethnicity and socio-cultural structure of the community, but this 1 in 700 more or less static. In any published literature and book, you could found this particular rate. Trisomy 21 is also the leading cause of spontaneous pregnancy loss. Perhaps all of you are aware about the fact that there are some instances when mother lost his pregnancy without any common reason, without any structural deformities, without any accidental deformities. And this is mainly due to the presence of this excess number of chromosomes in the embryo that ultimately selected by natural selection to be eliminated, and that's why the pregnancy is being lost. So first of all, this is a great man in 1866. He is a British medical practitioner or physician known as John Langdon Down.
Sorry to interrupt you, sir, but I guess the slide is not moving. Purnapamar, Salma, can you check?
No, only
Okay. I'm just doing that. Okay. Is it visible now?
It is visible, but not in full screen mode.
No.
The left panel is still visible.
See whether this is visible or not. Is this visible?
Yes. The second slide is visible.
Okay. Now come to this one. Is it visible now?
Yes.
Okay. So this is John Langdon Down, who first identified a group of children reported to his dispensary or to his chamber with a particular set of character, that same mongoloid face, that is the mongoloid race of the human. And he found that this group of children were absolutely idiots in terms of their intellectual ability. So he wrote a paper in 1866 and that entitled Observation on the Ethnic Classifications of the Idiot. This is the first published literature regarding Down syndrome. But that time he did not shed any light on the cause of this particular set of characters or cause of this intellectual disability. Nearly about 100 years later, another great man and British physicians who identified the cause of this particular idiocy or intellectual disability is nothing but the presence of extra copy of chromosome 21, and he described this condition as trisomy 21 in the year 1959. And this is the photographs of Jérôme Lejeune. This Jérôme Lejeune, the great man, identified firstly, this is actually the genetically backed intellectual disability, and presence of extra copy of chromosome 21 is the major cause of this kind of the problem. Next. This is the karyotype, you can see, and even these are very familiar. These all chromosome structures are very familiar to you if you go through any book, and you can see there is extra copy of chromosome 21, that is triplication, and this triplication is a cause of trisomy 21 and Down syndrome birth. But how this extra copy is originated, that is a big question. The error that happened during the meiosis, that is the process of cell division that led to gamete formation, is known as Nondisjunction. That means failure of homologous chromosome to separate properly during meiosis. If you compare what actually happened in the normal ideal condition and what actually happened during the error, you can go through this cartoon and confound. And the left hand panel, we can see the homologous chromosome. One is depicted in yellow, another in the blue, can be separated in the meiosis 1 ideally, and then their sister chromatid again can separate it into the haploid set of chromosomes into the gamete following meiosis 2. But in case of non-disjunction, the chromosome may separate properly at the meiosis 1, but not in the meiosis 2, and that results in the gamete called disomic gamete having two copies of haploid chromosome of a given chromosome that experience that error, and there is another gamete where there is no such chromosome. So this kind of gamete having two copies of a given chromosome can be fertilized with a healthy sperm, and that sperm also carry another haploid copy of this chromosome, lead to the formation of triplicated condition in the zygote, and this is the cause of Down syndrome. Well, if we go into the next slide, what happened in the normal meiosis, the chromosome usually pairs, and during the pairing in meiosis, they exchange their body part, that is known as recombination. As you all know, recombination is an essential phenomenon for generating variation into the genome of a given animal. But at the same time, recombination is important not only for the generation of the variation, but also for proper segregation of chromosome. If you look at this diagram, you can see these chromosomes, they tie up their legs, that is their arm, in the X-like fashion. And this X-like structure is known as chiasma, and that chiasma actually holds the chromosome tightly. On the other hand, there is a pull from the opposite pole, pull from this centromere towards this pole and pull from this centromere to this pole. So there are two counteracting forces. One force help to tie up this chromosome, and another force that help just to separate out the chromosome, and balance between these pull are important for separating the chromosome properly. So in meiosis 1, there is absolute proper separation of the two homologous chromosome, one in primary oocyte, another one is first polar body, and following meiosis 2, all the chromosomes get separated into the sister chromatid, and all the polar body receives haploid set of chromosomes, and the oocyte set receives the haploid set of chromosome 2. This is the condition what we see in case of normal meiosis. In case of meiosis 1 error, that means if non-disjunction happens in meiosis 1, then there is some problem in the pairing and recombination. I am coming to this story later on, and that led to the non-separation of the chromosome. That means both the chromosomes remain in the primary oocyte, and this polar body doesn't receive anything else. And following their separation, the oocyte receives two copies of the sister chromatid instead of one. We often call this phenomenon as meiosis 1 non-disjunction or meiosis 1 error. In case of the meiosis 2 error, the chromosome, homologous chromosomes separate healthy and ideally into the primary oocyte as well as into the polar body, but not the sister chromatid. There is some problem in the sister chromatid separation, and that sister chromatid separation problem led to the having two copies of sister chromatid in the mature oocyte. So these are two different kinds of mechanism of non-disjunction. One is meiosis 1 non-disjunction, we often called it as M1 error, and meiosis 2 non-disjunction as M2 error. Beside non-disjunction, there are some other mechanism of having trisomy fetus, and that is translocation. Often in about 0.02% of the female, the oocyte experience a translocation event between normal chromosome 14 and normal chromosome 21 that led to the formation of that kind of hybrid chromosome. So if oocyte receive this hybrid chromosome, that is translocated part of chromosome 21 on chromosome 14, and it has another healthy copy of haploid chromosome 21, and if that oocyte receives another third copy of chromosome 21 from the father side, from a sperm, that led to the partial trisomy 21 condition, and this partial trisomy 21 condition ultimately culminate into the development of a fetus with Down syndrome phenotype. We often call this as translocation Down syndrome type. In my lecture, I am not going detail about this translocation type of Down syndrome because this is a very minor group in terms of their frequency and occurrence in all the tested population across the world. Now the question is, following the discovery of John Langdon Down and the Lejeune, the entire scientific community, particularly the medical practitioners and physicians of the UK as well as some part of the USA, they started searching what is the cause of having that kind of the baby in certain group of women. So in search of that fact, the first identified risk factor emerged as advancing maternal age of conception. That means if a lady wish to have baby in their advanced stage, that means beyond the age of 32, 33, 34, there is a higher increase, a higher chance of increase in that kind of the incidence. And that risk factor was initially identified by Penrose, another British physician, in the year 1933. If you look at this graphical presentation, you can see in this x-axis there is maternal age group, that means women of the different ages, and on the y-axis there is different ages, and the x-axis there is a percentage, and you can see that aneuploidy goes high with the age, and there is a failure of implantation with age. So the fertility reduces with advancing maternal age, and that advancing maternal age is a big risk factor for having that kind of the non-disjunction, error in the oocyte. Probably with age, there is degradation of the protein machinery that ensure proper chromosome segregation, and that causes a higher risk among the older women to have baby with erroneous chromosome number. So here is some global statistics that shows how the frequency of Down syndrome birth go high with maternal age. At age 20, if women conceive, she has a chance of having 1 in 1,500 risk having baby with Down syndrome. At age 30, the risk is become little higher, 1 in 800. At age 35, the risk is 1 in 270. At age 40, the risk is 1 in 100. And at 45, the risk is 1 in 50. So you can see there is a gradual straight line increase or exponential increase of risk of having Down syndrome baby with advancing age of conceptions. From here, we started our work. These are something you can consider it as a prelude or introductions of my work. So we just started with some screening tests for the Down syndrome pregnancy. There are certain serological marker in the maternal blood that help us to monitor and anticipate regarding the pregnancy outcome, and we can anticipate, yes, that baby may have certain problem and may be developing into a Down syndrome baby. And what are they? The first of is called nuchal translucency in ultrasound scan. As you know that ultrasound scan is very common in the pregnancy conditions, and our ladies are usually go for ultrasound at least 3 times during the pregnancy. I can show you one ultrasound photographs. You can see there is a transparent area just beneath the head. This is the head of the developing fetus. And this translucent area is very common in case of the Down syndrome, but in case of the normal baby, this part is not translucent, and this opaque area actually fill this black gap in case of the normal fetus. But in case of the Down syndrome fetus, because this is a ultrasound photographs of the Down syndrome fetus, you can see this transparent area, and this is called nuchal translucency. It is a very strong indication that fetus is developing with some problem in their neural system. Beside ultrasound detections, there are certain serum marker And these are alpha fetoprotein. In normal condition, the alpha fetoprotein level in the maternal blood remain high, but if there is Down syndrome fetus in the mother's womb, the alpha fetoprotein level goes low. Estriol, the second one, is another hormone. As you know that it is also a good marker for detecting Down syndrome, because the estriol level also goes low in case of the Down syndrome pregnancy. Human chorionic gonadotropin, the hormone, another important hormone, that actually exhibiting higher concentration when there is a fetus with Down syndrome in mother's womb. Inhibin A, another important hormone from the ovary, that also exhibit higher secretion in the maternal serum following inceptions of Down syndrome baby. And another protein marker in the maternal serum that goes low, this is known as PAP-A. This is a very strong indicator of the Down syndrome when you will find it lower than the normal ladies in some women, because that time we can interpret, yes, this lady may have a pregnancy with Down syndrome. But that all the tests are actually predictive. We can ascertain their existence, that is existence of the free chromosome, in this technique called amniocentesis. You are familiar with this technique? In case of amniocentesis, what we do, we just take out some little amount of the amniotic fluid, because amniotic fluid carry some cells from the fetus. So if fetus is trisomy 21 genetic background, all these cells will exhibit trisomy 21 condition following karyotyping. So amniocentesis is nothing but the collection of the amniotic fluid, and to do culture with this fluid for amplifying the cells. And with this amplified fetal cells, we can go for classical karyotyping and chromosomal study to monitor what is actually happening at the chromosomal level. So amniocentesis is the highest level of as confirmatory test, you can say it, confirmatory test, from where we get an absolute 100% assurance, yes, the baby is growing with Down syndrome. There are some molecular tests with short tandem repeat marker, popularly known as STR marker or microsatellite marker. Perhaps some of you have heard this microsatellite term. In our BSc syllabus too, we have microsatellite. So microsatellite repeat marker are highly chromosome specific. So if you can use this microsatellite marker and PCR, that is polymerase chain reaction technique, to amplify the exact chromosome 21, and you can get this type of the photograph. Look at this right hand gel photograph. You can see this middle panel of the gel. It carries three distinct bands, and these three distinct bands are representative of three distinct chromosomes as fished out by the microsatellite marker. So microsatellite marker can bind with the different chromosomes, and following their amplification, they will produce that kind of the band in the gel electrophoresis. So three band means three distinct chromosomes. On the other hand, if you look at this right side panel, this is the genomic DNA of the father, and that exhibit two bands which are representative of the two normal chromosomes. And on the mother side, you can have two bands, and these are again representative of the two normal chromosomes. But the advantage of this technique over the classical karyotyping is that you can monitor from which parent the extra chromosome is coming. That means you can compare the band position, father, mother, and Down syndrome children, and you can see these maternal two bands are exactly matching with the two bands of the Down syndrome, and only one band of the father is matching with the Down syndrome. That means both the chromosomes of mother enter into the oocyte during the genesis, and so the non-disjunction error originates in the mother side, not in the father side. If it is from the father side, then both the bands of the father would be inherited in the Down syndrome, and mother will have only one band sharing with the Down syndrome baby. So in that way, we can interpret from which parent excess chromosome is coming. Then we started research with women having Down syndrome baby. Our primary aim or objective was to find out the risk factor, both the genetic risk factor as well as environmental risk factor that predispose means make the women more susceptible for the Down syndrome baby. Our working hypothesis was that non-disjunction event is not sporadic. Rather, non-disjunction event must have some prelude in the genome of the women. So some of the women may have certain genetic marker or genetic signature which make them more and more prone to have Down syndrome baby. That was our belief, and we started with this working hypothesis. So our key question was, first, how does recombination affect chromosomal segregation? Because that time when I started this work in the year of 2001 and 2002, that time one particular notion was standing, and that notion was, if there is some anomaly in the recombination, that may affect chromosome segregation too. As I told you earlier in my earlier slide, that recombination has some positive role in proper chromosome segregation beside creation of the genetic variation in the genome. So we started with this working hypothesis. Probably there is some anomaly in the recombination event in the chromosome, and that led to the non-disjunction. And our second question was how recombination frequency and pattern interact with maternal age of conception. Because as I told you the story, the first identified risk factor was advanced maternal age, and it was recognized by the Penrose, the British medical practitioners. So if recombination is a risk, if advanced maternal is a risk, so at some of the point of oogenesis, there should be some interaction between the recombination and maternal age. So this is our query, how recombination and maternal age work together to make the oocyte more susceptible for chromosome 21 non-disjunction. So what we did, we selected a panel of STR marker, that is short tandem repeat marker, spanning the entire long arm of the chromosome 21. You can see we had segregated the entire chromosome arm in almost 6 equal interval, and these are the base pair number and interval margin. And these are the set of panel of STR marker, and with this marker we start genotyping. How? Using this satellite marker we start doing PCR for maternal, paternal as well as Down syndrome children, and we looked into the inheritance pattern of the allele, as I exhibit you the previous slide, where you can see how the bands are coming from mother side and father side. For each and every microsatellite marker, we did the same kind of the gel electrophoresis, and we followed the same kind of the band inheritance pattern in that way. Just coming to the same photograph again to understand how we interpreted the recombination. So these are the amplification of the same family, PCR amplification of the same family, but using different microsatellite marker. In one microsatellite marker, we can see that both the DNA bands of the mother is in toto inherited to the Down syndrome baby. But if you look at the left panel, you could find that Down syndrome baby basically showing two band, but one band is more intense or almost double in concentration than the other band. And these band, the intense band, is matching with one chromosome of mother. That means maternal one chromosome get duplicated during the meiosis, and then duplicated copy moved to the Down syndrome children. But here this duplication did not take place, and two different chromosomes of the mother entered into the Down syndrome baby. So we describe this condition as non-reduced. That means the heterozygosity of the band, if you consider these are heterozygous condition, two distinct bands are there. Heterozygosity of the band was retained in the Down syndrome. So we called it as non-reduced condition of the marker status. And if there is a reduction, that means two bands, two heterozygous condition of the mother is actually becomes homozygous. That means one chromosome of the mother get duplicated during the movement into the oocyte, and we called it as a reduced condition. So in that way, we interpreted the genotype of a given family like N, 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 N. That means non-reduced, non-reduced. If you go for genotyping with a distinct marker sequentially, say for example, for the first marker, you got non-reduced condition. For the second one, you got non-reduced. For the third one, you got non-reduced. For the fourth one, you got non-reduced. But for the next one, you got reduced status. That means status like that. Whenever there is a change from non-reduced to reduced or reduced to non-reduced in a given string of marker for each family, you will interpret this transition as recombination event. You may ask me the question, sir, what is the basis of this interpretation? Yes, that basis is basically a statistical interpretations. I'm not going detail into that because that will take another lecture of the 2 hours to make you understand how statistically we infer what is recombination, what is not. I'm just telling you the outcome of my study. So in that way, we go for genotyping of the each family. I am showing you a small partial data of thousands of the family data that we have in our lab. And this left side panel of the Excel sheet representing different families. And each string of this family is actually showing the genotyping data of the successive marker. And at the top of this panel, you can see different color which are actually representing the different markers, microsatellite marker. So if you go in some marker emerges as uninformative, you cannot interpret the origin of the extra chromosome in the baby. And some marker are informative, again informative marker, maybe non-reduced or reduced, and we look for where actually this transition happened, and we scored one recombination event at that point. In that way, we go on scoring this recombination event for the thousands and thousands family. Till date, we have nearly 1,500 family recording that way. So I'm showing a small part. After getting this information, raw data from the gel electrophoresis, we put this data into a software known as NDGMAP software. It is another statistical software based on Bayesian statistics known as expectation maximizing algorithm. I am not a statistician. I don't know the algorithm very well. That software was designed by my collaborator and friend, Professor Eleanor Fingold. I will show you the photographs. She is presently the Dean of Pittsburgh University Science Department, and she is a geneticist as well as statistician. So she did favor for me and designed this software in which I have just put all my raw data of the gel electrophoresis and got results in that way. You can see these digits. Don't go for digits in detail. Just look at this last panel. This last panel actually representing the recombination fraction. So through this statistical software, I have processed the entire gel electrophoresis data to get the recombination fractions between the marker. What was the ultimate outcome? Well, look, till date, though this number has been increased right now, nearly we have 1,500, but the time that I prepared these slides a few months ago, we have 1,032 patient family. We scored nearly 92% of the all cases, the error originated in the oocyte. That is, we called maternal error. That means the women are more vulnerable to experience that kind of the error in their oocyte. Only 8% error originated in the sperm. So males are not prone to non-disjunction error due to some of the reasons. I am coming to this point later on. Among the all 92% maternal error, if we segregate them again as a meiosis 1 error and meiosis 2 error, we scored nearly 70% of all reported cases of maternal error have their problem in meiosis 1, and only 20% cases the problem originates in meiosis 2. And there is a small fraction of post-zygotic error. That means the non-disjunction happens after fertilization. During the course of fertilization, the embryo was absolutely normal and euploid with correct chromosome set, that is 46 chromosome number. But following rapid gastrulation, blastulations and cell divisions, error may arise mitotically, and that score only for the 3%. On contrary, among the 8% of the all paternal error, we again segregate this paternal error in the meiosis 1 and meiosis 2 group and found 42% of the all paternal error is actually originate in the meiosis 1 phase and 58% in the meiosis 2 phase. That means for the paternal error group, almost comparable frequencies were recorded for meiosis 1 and meiosis 2 category. So we started working with this group, that is maternal group, because this is the largest group having problem. Paternal group may have some sporadic problem, so we focus on the maternal group. So what we did, we scored recombination as 0 recombination, 1 recombination, and 2 recombination, and these recombination frequencies were segregated according to the maternal age of conception. We segregated all participating mother into 3 age group. Firstly, the young age group who had their age of conception of Down syndrome baby less than 29 years. Then middle age group who had the age of conception of the Down syndrome baby 29 to 34 years. And the old age group, that is the women having Down syndrome baby over the 34 years. So we have 3 functional age group. We categorize them in that way for our statistical interpretations and statistical testing. So following these statistical distributions, we go for making that graph. I'm not going detail into this graph because these are some part very critical to understand. I'm just going to our hypothesis. What we hypothesize from all these statistical data and noise? Yes, we found 3 different recombination patterns which are proved to be susceptible for chromosome 21 non-disjunction. The first group is no recombination at all. That means if the chromosome 21 in oocyte does not participate in recombination during meiosis 1, that pose a big risk of having non-disjunction in meiosis 1 among the younger mother group. Just recall again, Penrose told that advancing maternal age is the risk of non-disjunction. But during the course of sampling and study design, we received lot of women who had Down syndrome baby at younger age, that is less than 29. The age which is basically a very fertile age for having a healthy euploid baby. But that particular group accidentally got Down syndrome baby in their ideal age of reproduction. So that is very surprising, and we focus our interest on that particular group. And we found that this young mother may have Down syndrome baby if their chromosome 21 did not participate in recombination process. So no recombination is meiosis 1 error among the young mother. Then we found another category of recombination which is very close to the telomeric end. That means recombination at very close to the telomeric end or peritelomeric area may create some problem in chromosome distribution and lead to chromosome meiosis 1 non-disjunction again among the younger mothers. And we identified the third category of susceptible recombination that is very close to centromere. if there is recombination very close to centromere that leads to chromosome entanglement. That means chromosome becomes spirally coiled to each other and they could not separate out. And that led to meiosis 2 non-disjunction at older age mother. Usually, in normal cases, in case of the birth of a normal euploid baby, recombination takes place somewhere in the middle of the chromosome arm. The middle of the chromosomal recombination event is ideal recombination event for chromosome segregation. But we found no recombination is a risk of chromosome 21 non-disjunction at younger age of mother at meiosis 1. We found peritelomeric recombination, which is again risk for meiosis 1 non-disjunction among the younger group. And thirdly and lastly, we found pericentromeric recombination pattern, which is risk factor of meiosis 2 non-disjunction at older age group mother. So we just go for publishing this data, and it was accepted in the American Journal of Medical Genetics. We proposed our hypothesis. You can see there is Eleanor, because unless Eleanor, we could not able to decipher this complicated genetics of this recombination and its relations with the maternal age. And successively, we have two publications. That was my initial work in 2009, followed by publication in Cambridge, genetic research from the Cambridge. And this is also the confirmatory to the previous notions that chromosome 21 non-disjunction basically involve certain degree of anomaly in the recombination pattern. Okay, so what next? Well, keep in mind the fact that there are a lot of events and citations across the world that if there is some excessive pollution, you know, Chernobyl disaster, you know, there are a lot of such kind of the disaster happening in the world, and following this disastrous event, there is an just outburst of the Down syndrome baby in the next few years. That means environment has some strong deleterious effect in the oocyte maturations and oocyte formation. Keeping this fact in mind, we started searching, apart from the genetic factor, is there any environmental factor or habitual risk factor that may increase the chance of non-disjunction in the maternal oocyte? So through our epidemiological recording, because during the course of recruitment of the family, we have a big data sheet. And within that data sheet, there are about 100 questions. We ask those questions to the family member, that is, these parents of the Down syndrome, regarding their lifestyle, regarding their practices, regarding their food, regarding their livelihood, regarding their exposures, regarding their other diseases. These are very important parts. Even you people, when go for such kind of the epidemiological study, you need to prepare that kind of the questionnaire, long questionnaire, to find out some relevant informations from that. Well, so following this, we just sorted out what are the very common among those families, and we found two important factors. One is chewing tobacco practice in our society in the different forms of gutka, jorda, and everything from the early days of their teenage. And the second one is irregular contraceptive pill use. That is dangerous. I'm coming to this point later on. Why irregular contraceptive pill use or emergency pill that are very usual in our daily life. We can see that kind of the advertisement in the televisions or different channel that they are talking for the emergency pill use, but these are dangerous for the women body. I'm talking this story later on. So again, we go for different statistical modeling. Actually, you know, the pure genetics or classical genetics basically dealt with statistics. Without statistics, genetics cannot be done. Starting from the Mendel's day, Mendel was a statistician, you know that. Mendel's day till date, we the geneticists basically depends on the statistics heavily. We did molecular biology a little, but through the statistical processing, we just extract out the hidden informations coming from the molecular biology outcome. And that is the beauty of the genetics. Again, we started different statistical modeling, and I'm not going again detail into that. I just will focus on the outcome of my results. I'm just showing these statistical tables. So there are number of the digits, number of the software, and from that we extracted. Hello? Is there any problem? Someone is talking.
No, sir. Please continue.
Okay. Okay. So through this statistical modeling, we just came out with our notions and new hypothesis that epidemiological Down syndrome, new insight into the multi-dimensional interaction among genetic and environmental risk factor in the oocyte. That was a very big publication in American Journal of Epidemiology in the year 2011, where we told that chewing tobacco is a big risk factor for absence of recombination. So we found that mother or women who experienced chewing tobacco habit very early of their teenagehood, they actually suffer from the lack of recombination. It may be possible that chewing tobacco somehow disrupt the protein machinery in the oocyte, which are important for the chromosome segregation. So that was a big publications from our part. Now, importantly, one of my colleague, she is now in the USA, asked me that, Sujata, could you look into the molecular aging? That suddenly came in her mind at night when he went with some concept of the telomere, because that time telomere paper came first, and we hope that telomere will emerge as a very important marker for the all aging related disease. So I am just coming to her name. She is Shumita, is a brilliant guy. She suggested me, Sujata, you can look at molecular aging of the mother. So I started thinking for that, and entire team we started working on estimating the telomere length of the Down syndrome mother. So our working hypothesis or anticipation was that, because whenever you will start one work, we must have one belief in your mind that that may be the outcome. So that is your hypothesis. So our hypothesis was it may possible that if you take two groups having 30 years age and 30 years age, both have a chronological age in terms of year, 30 year, but one group may suffer from silent molecular aging, but another group is not. That means in one group there is rapid degradation of the protein machinery, rapid degradation of the hormone, rapid degradations and deteriorations of microenvironment of the ovary, etc., which could not be discernible for the outside. From the outside, we could not identify how is going into the ovary. But another group may not suffer from that. So we took out the DNA sample from these two groups. One group having age 30, having Down syndrome baby. Another group having age 30, but have absolutely normal euploid baby. And we just go on cutting the telomere length with specific restriction enzymes, and then ran the gel electrophoresis and found this photograph. You may understand it's very easily. You know that any DNA sample which have larger fragment size may retard into the gel. They move slowly into the gel and remain stacked at the top of the gel. But the DNA sample, which gets fragmented into small pieces, usually ran faster and move forward through the gel. So we just go on comparing mother having downs and mother having no downs. So you can see that mother having no downs have larger DNA fragment from their telomeric end, but mother having Down syndrome baby having smaller telomeric DNA fragment. That was the first discovery from our part in the world. We were the first who advocated that mothers having Down syndrome children may suffer from molecular aging, which can be estimated from their telomere end than the mother of the same chronological age but having euploid baby. Am I clear to all? So we have that kind of discovery. So again, we go for estimating what is the rate of telomere shortening among these group, those who have euploid baby and those who have Down syndrome baby. We found in case of control, that means mother have euploid baby, experience only 61 base pair shortening per year. In contrast, mother having Down syndrome baby suffer from rapid telomere shortening, and it's about double, 110 base pair per year. Okay, that is a big discovery. So we just go on publish this data, and it was accepted by Human Genetics from Germany, another very elite journal in that field. And we make a title, catchy title: telomere length is associated with type of chromosome 21 non-disjunction, new insight into the maternal age effect of the Down syndrome. But because before us, everyone talking about the chronological age, that is age of 32, age of 34, age of 35 is risky age for having Down syndrome. We for the first time told that no, this estimation of the age in terms of the year is not enough to interpret, because some of the younger mother, we saw that they have Down syndrome baby. That means even if they have the age less than 28 years or less than 24 years, but inside of their oocyte, inside of their ovary, inside of their body, there is rapid molecular aging. That's why they are suffering from non-disjunction even at the so-called lower chronological age. But again, scientific mind is very inquisitive, you know that, and whatever we told cannot be taken granted by the Western world. So our collaborator Stephanie just go on replicating the same study. After publication of my work, Stephanie called me and he just replied, Sujoy, what you did, I am interested to do the same experiment on the US population. So you can look at this a year, that is, we had a publications in the 2010, and from 2010, Stephanie's group started working on the Caucasian population, the same experiment that I did from my mind. He just replicated the entire experiment on the Caucasian population, and very surprisingly, and it was a reward for me, because they came to the same conclusion and published their paper in 2015 in the same journal, that is Human Genetics, received their journal, received their manuscript as supportive to their earlier notion that I published into the same journal, and their title of paper was Association between Telomere Length and Chromosome 21 Non-Disjunction in the Oocyte. Almost similar type of title, and these publications from USA was confirmatory to my novel and pioneer publications. And after getting these two publications together, now the entire world start believing, yes, some women may suffer from molecular aging, and if they suffer from the molecular aging, they are at the higher risk of having Down syndrome or any other kind of the baby with chromosomal error. Okay, well, now move on to the next part. So we hypothesized that mothers of the Down syndrome are genetically older than the mothers of the normal children at their older chronological age, and in some way the molecular apparatus of telomere maintenance is linked with molecular apparatus of the chromosome segregating system and pattern of non-disjunction. That was our notion. Now we started thinking again, what is the relation, or is any relation exist between the chromosome telomere shortening, that is rapid molecular aging, and tobacco use? Is it there? That was a big question that came in my mind. Is there any relations of chewing tobacco use that we described in our 2011 paper onto the telomere shortening, that is rapid molecular aging? Again, we go for that kind of analysis, go for extensive statistics, statistical modeling. I'm just skipping these slides. You can see at this, these are nothing but the statistical jargons. These are very, very boring, I think so. But the finding is important. Whatever the statistical jargons and noises that we practice ultimately led to making some story, and this is our story. What we find that time, smokeless chewing tobacco and shorter telomere length are associated. That is very, very, very pioneering hypothesis from our end. Because before us, nobody in the world told these notions very strongly. We exhibited that somehow practice of the chewing tobacco accelerate the maternal molecular aging. Well, and these tobacco is maternal age independent risk factor. That means the lower age group mother as well as older age group mother, whenever they use tobacco, they may suffer from the risk of non-disjunction. And it was done by first scholar Anirban, and this is the paper came in the Public Health Genomics in 2015. And the title was Maternal Telomere Length and Risk of Down Syndrome, Epidemiological Impact of Smokeless Chewing Tobacco and Oral Contraceptive Pill on the Segregation of Chromosome. So up to this point, we dealt with recombination anomaly, telomere shortening, smoking, contraceptive pill use, etc., etc. Now we start thinking why the recombination is anomalous in certain women. Is there any genetic marker? Is there any genetic predisposition? So to get insight into that, we started candidate gene analysis. That means we go on searching literature, what are the genes that regulate recombination. So we found a long list of the genes. And from that list, we just selected out some of them, keeping their impact onto the recombination event. So our first choice was PRDM9. This gene is very important, not only for the human being, but also for the entire ape group. That means the gene was originated early in the ape evolution, when we all were ape. And this gene almost conserved in its structure and function throughout the primate group. So PRDM9 is actually act to specify the location of recombination onto the chromosome. The where exactly to which place of the chromosome recombination will take place is actually determined by the PRDM9. So as we saw that there is some problem in the position if recombination takes place more close to centromere or more close to telomere, may be error prone and may lead to non-disjunction. So keeping this fact in mind, we chose PRDM. Our working hypothesis was if there is certain mutation, if there is certain polymorphism in PRDM9, that will disrupt the optimal functioning of the PRDM9, and that may lead to erroneous placement of chiasma more close to centromere or more close to telomere. So we're searching PRDM9 reading frame for new mutations, new polymorphisms through the dideoxy sequencing, Sanger sequencing, and found 3 distinct mutations. This arrow mark is nothing but the new mutation that we found onto the reading frame of the PRDM9, particularly from the mother of the Down syndrome baby, not from the mother of the euploid baby. All the mother of the euploid baby did not exhibit any kind of the mutations in the PRDM9 reading frame, but the mother of the Down syndrome baby exhibit frequent occurrence of these mutations. Beside PRDM9, we look for the another gene that is presenilin-1. Presenilin-1 is a particular protein which helps in chromosome tightening and chromosome ligations in spindle fiber. And it helps in chromosome segregation. It is very important for the anaphase movement of the chromosome too. So our working hypothesis was that if there is any mutation and polymorphism that disrupt the PSN, presenilin 1 functioning, that may lead to improper chromosome segregation at anaphase. And again, our intuition proved true, and we found certain mutation among certain Down syndrome bearing women where we saw there is a transition of T2G and T2C in the presenilin 1 that could disrupt the functioning of the presenilin. And another important gene, the third one, is MTR. MTR is basically a gene which regulates the folic acid metabolism. As you know that folic acid is very important, and doctors usually suggest to take folic acid during the course of pregnancy because it helps in healthy making of brain and neural tissue in the developing fetus. But we hypothesize that folic acid is equally important for oocyte development before baby formation, because folic acid acts as a methyl donor, that it donates methyl group, and that methyl group binds with histone at the centromere, and this methylation of the centromeric histone is important for the proper segregation of the chromosome at the two opposite pole. Well, so we started looking at different genes and their mutations from the folic acid metabolism pathway, and again found, yes, some of the mother, there is mutation A2G in the MTR gene, and again in the control mother there is no such mutations. So in that way, presently we are searching some other gene too. I am not showing all the data because they are under the process and they are in the process of publications. So whenever it will be published, it will come into the website. So in that way, we just go on searching candidate genes among the maternal genome, and we hypothesize that the occurrence of non-disjunction and subsequent Down syndrome, but it not as an accidental or sporadic event. It may be sporadic and accidental in case of the environmental influence that we saw in case of tobacco use or contraceptive pill use. But some of the mothers, some of the women may suffer from this genetic predisposition. We often call it as genetic predisposition, means they are susceptible to have that kind of the problem, whatever their age may be. Even if all this mutation present in a particular woman, she may have Down syndrome baby at age of 18 or 19 of first motherhood. Whenever she'll opt for the motherhood at the first time, she will have Down syndrome maybe, if that kind of the polymorphism or mutation present in her genome. So in summary, we can say, if you look at the entire trajectory of the oocyte maturation, there are certain time and there are certain space of risk factor association. When there is a small follicle start journey in the oocyte ovary, there is a problem of the recombination as well as reduction in the recombination. When there is a puberty and up to the menopause, there is problem of the accumulation of the environmental insults. As the women exposed to the environment, different environmental risk factor, pollutants, everything enter into the body, and through blood circulation, ultimately they reach up to the level of the ovary, and within the ovary, within the developing follicle, they occur over the time, and they just try to disrupt the entire optimum system of the chromosome segregation. Okay, and ultimately that lead to the ovulations of an ovum with erroneous chromosome number. So all these smiling faces as a very big scientific story in the background that I have described you. Besides that, there are several other epidemiological risk factors. Obviously, maternal habit of the smoking, though smoking is not at all very prevalent in comparison to the Western world. In our country, chewing tobacco is very challenging, but smoking somehow, obviously, today's updated girl and the modern girls, they are familiar with the smoking. Even I, every time see them in my campus, that girls are taking smoke, taking puff, and I just call them not to have that because that will disrupt your oocyte formation. The second one is maternal habit of the alcohol consumption. Again, that is very, very harmful. Thirdly, exposure to the different pollutants and irregular contraceptive pill use. I'm coming to this point that irregular contraceptive pill is nothing but that these are actually the analog of different hormones. So whenever you are taking contraceptive pill without consultation of a physician or gynecologist, it will abruptly change the entire hormonal environment within your ovary. And these hormones, that is estrogen, progesterone, and their proper balance and titer are important for the proper chromosome segregation as well. They sends the signal for the meiosis, completion of the meiosis and chromosome segregation. So if you disrupt that delicate balance just by putting excess load of hormone analog in your body, that will disrupt and lead to chromosome segregation, irregular chromosome segregation. So these are certain epidemiological risk factors. I'm just showing some of the photographs to which our ladies are exposed, that is carbon monoxide. In the rural area, these practices are going on, and this fossil fuel somehow eliminate huge amount of carbon monoxide that may disrupt the oocyte formation. Then this practice, you are familiar to that. I spent 7 years in Sundarban College, and during my journey in the Sundarban, I saw this photo every time. The women are searching for the fish seeds, submerging their lower part of the body into the river, and these rivers is actually traversed by certain boats. This is the usual means of communication, and that boats release lot of oil spill. These oil spill ultimately absorbed in their skin, and that led to different kinds of the problem, including abnormal baby birth. And practices of smoking, you can see this photo. The grandmother taking the grandson on their lap, but taking bidi. And obviously the cigarette smoking. And this is party time with adequate amount of the alcohol. And you are allowing alcohol in your body during the cheer time. And obviously contraceptive pill. So up to that point, I am talking about the risk factor of the Down syndrome. But now coming to the second part of my lecture, that is COVID-19. This is the small part of my lecture, the recent development, recent risk that Down syndrome babies are facing. You know what is COVID. I am not going detail into that because all of we are very much expertise in COVID right now through our daily newspaper and media. So what happened with the Down syndrome? So there is an mRNA known as IFNAR1. This IFNAR1 mRNA is located on the chromosome 21. This chromosome 21, as Down syndrome baby has triplication of the chromosome 21, so they have excess copy of IFNAR1 mRNA molecules. These IFNAR1 molecules are actually the signal for the cytokine secretion from the WBC. So Down syndrome baby usually have excess amount of IFNAR1 mRNA that lead to excess secretion of cytokine. If you go for looking into the different serological parameters, cytokines and immunoreactive parameters, these blue boxes or gray boxes, whatever, blue boxes, it represents the Down syndrome baby condition, and these light gray boxes represent the condition of the normal baby. Look, this is CRP, cross-reactive protein, that remain high in case of the Down syndrome baby, but low in control. IL-2, interleukin-2, remain high in the trisomy 21 Down syndrome baby, but low in case of the control. IL-10 is too high in the Down syndrome baby, but low in the control. IL-6, TNF-alpha, IP-10, all these interleukin molecules and immunoactive agents remain in a high titer in the serum of the Down syndrome individual than the control. So what is actually led to? Don't think that excess amount of interleukin is healthy for excess immune reaction, but the story is absolutely reverse. That means excess interleukin and excess amount of all these immunoactive agent led to suppression of WBC, because these excess amount of the cytokine uh, gives always a wrong signal to the WBC for their proliferation. So without undergoing proliferation, WBC just go on suppressing their all activity, and there is immunodeficiency in case of the trisomy 21. We often, you can see this cartoon, the trisomy 21. So there is 3 copies of this IFNR1 and other genes, related gene. So IFNR overexpression led to the hypersensitivity, that is excess secretion of the different cytokines from the WBC, and this increased cytokine production by the immune cells led to depression in the WBC activity, and that led to immunosuppression in the Down syndrome baby. So Down syndrome baby not only susceptible to the COVID infection, rather they are susceptible to other kind of the respiratory infection as well. So usually if you look at the Down syndrome baby and ask his mother whether he is suffering from respiratory problems or any kind of asthma and everything, she will answer yes, because in general Down syndrome have immunosuppressive phenotype, and this excess secretion of the cytokine is known as cytokine storm. So cytokine storm is responsible for make this Down syndrome baby susceptible for the COVID infection. Keeping in this mind, we, the global team from basically from the USA and some scientists from the Europe, they constituted a task force to monitor the health condition of the Down syndrome across the world. And somehow, through my collaborator, they came to know my name and they included me in this task force only from Asia. So I am the sole representative of from the Asia in this task force, and we started working from last March, 2020 March. And after joining this group, task force group, Down syndrome task force group, we just go on taking a survey virtually, and we dissipated all these questionnaire form and epidemiological detail through NGOs and all other groups, and we succeeded to collect a huge amount of the data regarding Down syndrome. The first slide, this is some of the small outcome of our study. Now, what are the symptoms that are common in the Down syndrome patient during their COVID infections? So fever is the most common, followed by cough, shortness of breath, nasal signs, and everything. There are so many different ways. Next one is the mortality. If you look at this, the mortality rate of Down syndrome, this red bar is actually showing the Down syndrome.
Sorry to interrupt you. Sir, we have only 5 minutes left.
Okay, I'm just completing that.
Okay.
So these are some of the outcome regarding mortality, and these are some of the risk factor which make the Down syndrome more susceptible, among them Alzheimer's disease and mental dementia. These are some important factors that causes higher infection of SARS-CoV. And we made report in the different 14 languages, global languages, and I succeeded to put Bengali in them, Bengali and Hindi in them, along with Spanish, Russian, Chinese, Japanese, and so many others. And this is our global task force from the different countries, and she's Stephanie, my collaborators, and all these are working together. There are some social aspects of the Down syndrome that you should keep in mind, and you try to dissipate this concept among the other people, that family and parents having Down syndrome are stigmatized due to the lack of knowledge. Down syndrome children are not abnormal. We often regard them abnormal child, but they are not abnormal. They suffer from certain developmental and mental retardations. They are able to carry daily work and they can achieve success. Here are some success stories. These are successful Down syndrome from abroad, but Today, we received some information from the Indian Down syndrome who successfully completed their degree. They came into their professions. So this is Paul Soca, awareness leader, Pablo Pineda, Down syndrome patient, Spanish actor, Angela Bachelor, Spain counselor of the Spain, Tim Harris, largest KFC restaurant owner in California, and we observe World Down Syndrome Day in every March 21st. So this year also we are going to celebrate Down Syndrome Day by making one celebration events. And this is my research group. He is Anirban, my first scholar, followed by Upamanyu, Pinku, Upamanyu, and so many other researchers. This is Stephanie and Eleanor. I am talked about Eleanor, the statistician as well as geneticist who designed all the statistical model for me and my large research family at Science College. And lastly, every week we meet with Down syndrome family to make them concerned about the latest development of the research and how this research could be implicated to make their life more beautiful. So that's all. And this is my collaborator. This is Mara from Spain, Eleanor from Pittsburgh, Stephanie from Emory University, Andre from London, and my sir, Shubhrata Kumar De, who happened to my research supervisor, Tiffany Oliver from Spelman College, USA, my wife, Papia from BKGC College, Dr. Apurva Ghosh. He provided a huge number of the Down syndrome sample, Dr. Shupratim Sharkar from SSKM Hospital, and so on. So that's all about my story, and have a sweet day. Thank you.
Thank you, sir, for these wonderful sessions. Student participant, if you have any queries, you can ask our speaker in the chat box. Sir, can you take some question here?
Would you please read out the question for
Yes,
me?
yes, sure. First one is, what the difference between meiosis 2 and mitosis decision? I think
it would be disjunction.
yes, maybe. Kaushik Shaduk, are you listening? You can unmute yourself and ask question directly. I think he is not. Next one, what is the perfect age for pregnancy to prevent the chance of Down syndrome?
Sure. 22 to 28 or 29 is the ideal age when all hormonal parameters and all proteins, those who involved in the healthy oocyte making, remain at the optimum way. But still, as you saw in my slides and my findings, that Down syndrome may happen in the young age too. So these are actually the chance or probability. So there is less probability if you have child at the early age of the women, and if you have the higher probability of for that kind of the child in the latter age of the woman.
Next one is, in families of children with Down syndrome, is there a binary interaction between children and parents?
Sure. Because in some family, the Down syndrome children are being neglected, particularly in the lower strata group, socioeconomic group. But in the higher socioeconomic group, in our country too, the parents are coming and interacting with us, and after knowing the exact fact that Down syndrome children are not mad, they are not lunatic, and they started training their children just by sending them in the different training school, rehabilitation center, and children are doing very well. Down syndrome children can play music by participations and inspiration from the parents. So always there is a strong interactions. Whenever if you see any Down syndrome patients are very healthy, you just believe that their parents are actually taking care. So that interactions is important.
Okay. Is there any risk of having Down syndrome baby associated with polycystic ovary syndrome?
We did not find any kind of associations. Rather, we did not conduct that kind of the study. Yes, this is very interesting, and it should be in my mind to look into this fact. Maybe actually polycystic ovary syndrome may disrupt certain hormonal balance, and that may lead to chromosome segregation error. It's possible.
Next one is, what is the balanced translocation carrier woman?
Balanced translocation means if the part of chromosome translocated to the other chromosome, but the rest part remain there. There is no loss of chromosomal material. That is called balanced translocation. Okay. So if there is balanced translocation, then there should not be any, say, expression of that Down syndrome type of phenotype.
So there are a lot more. Can we go? Oh,
So sure, if you have time, you can ask. It's up to
Okay,
you.
we have some minutes. So, how cancer is related with Down syndrome?
Yeah, Down syndrome patients are susceptible for the leukemia because there is a gene called GATA4, and GATA4 is located on chromosome 21. So triplication of the chromosome 21 leads to triplication of the GATA4. That's why Down syndrome patients are prone to develop leukemia symptoms in the later of their life at the age of 25 and beyond. But very surprisingly, Down syndrome patients, they are resistant to any kind of solid tumor development. They don't have any solid tumor. Till date, we didn't get any informations that Down syndrome patients have solid tumor. Maybe they have certain genetic differences than the normal population due to triplication of this chromosome that suppress any kind of solid tumor formation.
Next one is, are there any physiological differences that could lead to stable isotope difference in tissue of those affected by Down syndrome?
I could not get the question. The meaning is not very clear of that question.
Okay, we can move to the next. Can hormonal imbalance be the cause of Down syndrome?
Sure, I told you, na, in case of that emergency contraceptive pill disrupt the hormonal balance and that lead to the Down syndrome or any kind of the syndrome like that.
How to measure the topic specific vocabulary acquisition in children with Down syndrome?
Well, this is not my domain. What I can suggest that this is actually done by the advocator and trainer, those who are involved in the training of Down syndrome children. And yes, there are certain strategies. They have certain instrument and skill development regimen through which the students or Down syndrome students get benefited and they learn a lot of things.
Can we prevent molecular aging? How?
No, there is no way to prevent molecular aging. What we can do, we can retard the molecular aging by healthy lifestyle, healthy practices, not to smoke, not to take alcohol, minimum exposure to pollutant, and obviously taking a lot of antioxidant. This is usually we have to go for antioxidant taking in terms of fruits or any other healthy fruit.
Okay, and last question is, as telomerase activation is associated with cancer, can we slow the rate of cell aging by activating telomerase without the risk of developing cancer?
That can be done in the era of gene manipulations and gene targeting. You can design some synthetic molecule that is synthetic oligo to suppress certain genes or proteins that are associated with telomerase. So selectively you can augment the activity of the telomerase, increase the activity of the telomerase enzyme in specific, suppressing the other component as well. But that needs an elaborate molecular, this is something hypothetical that I'm telling. But if you want to materialize this entire concept, you need to go for detailed molecular experimentations.
Okay, I think there is no more question. Student, participant, if you have any question, you can unmute yourself and ask sir directly.
Excuse me, can I ask one thing?
Okay, ask Shreya, ask.
Yeah. So basically, you said the context of the molecular aging and you compared that between two means of the same age, right?
Yeah.
So did you see any physiological difference between both of them? Means external
Apparently not.
physiological difference?
Shreya, apparently not. But what I predict that physiological differences may be there. If you go for taking account of oxidative stress in the WBC, peripheral WBC, perhaps that will emerge as one of the indicator of the molecular aging. But we did not look at this because that time our focus was on the molecular estimations of telomere. So this is a good suggestions. Anybody could take this as a next project or next venture for the research. It's very simple. There are a lot of technique, simplified technique, and that will not cost more. That just look at the oxidative stress in the WBC. These are physiological parameter, and taking from mother having euploid child and oxidative stress in the WBC of the mothers having Down syndrome child. And I sure that there should be some difference.
Thank you very much, sir.
Okay.
Sir, can I ask a question?
Yes, yes. Okay, you
Sir,
can.
so you told that if we have OCP, then it have a chance to have Down syndrome baby. If the hormonal imbalance is gained, like I am not having OCP, but I have high thyroid or high prolactin,
Yeah, yeah,
so it can it cause Down syndrome baby?
yeah. That may, because if you go detail into the hormonal regulation, endocrine regulation of the gametogenesis, you can see other than, in addition to estrogen, progesterone or steroid hormone, almost all hormone, and thyroid has a very basic involvement in all kind of physiological activity in our body. So what you can do, is my suggestions. If some of you have some problem in thyroids or any other hormones, you just, beside the treatment, you just go on healthy practice of exercising. That is the only way. If you go the physical exercise every day, at least for the 40 minutes, lot of problem can be mitigated. Without taking drug, anyone can mitigate this problem, resolve this problem to a great extent by exercise, by yoga and everything. So physical exercise is important for all kind of the problem.
Okay, sir. Thank you.
Okay, we are not taking.
Sir, can I
No.
ask one question?
Yes, sir. Sir, go on, sir.
Sir, good morning, sir. I am
Good
Odhiraj from
morning.
Sri Vidyashagar College. I want to ask you is how reliable is USG in detecting Down syndrome?
Is very reliable. But one thing is very unfortunate. I am not going say anything against anybody else, but if the USG plate is read and interpreted properly, the indication of the Down syndrome can be perceived at the very early stage. But what happened? We have the USG plate in my hand, but we could not read out the content. That is the tragedy in our country. USG can detect the Down syndrome. At least that indication, that is nuchal translucency, length of femur, length of tibia fibula, all these can be measures from the USG plate. And all these parameters, when go some beyond the normal level, suggestive of having any problem in the fetus, maybe Down syndrome or any kind of symptom. But what happened, that our physician friends or medical practitioners, they, I think they perceive all these facts, but they would not say anything that time. But it can be interpretable.
How many weeks? Minimum, how many weeks should the fetus be to detect all those things?
If you go taking USG, second USG at the 4 month or 4 month, yeah, 4 month USG is the first stage when this kind of the problem can be detected.
Okay, probably that may be one of the reasons why our physician friends are not speaking anything about it, because in our country, the rate of abortion would increase.
Yeah, that may be cause. That may be the cause.
Okay, sir. Thank you.
Thank you, sir. Due to our lack of time, we are constricting the question hour session. Thank you, Dr. Sujoy Ghosh, for being with us and sharing a knowledgeous topic with us, which is very much interesting for us to participants and students also. Plus, sir, if you have some time, please be with us. We have a short valedictory session.
Sure.
Shalma ma'am, over to you. Are you there?
Yeah, I am here. Okay, we're at the end of our international webinar. We are just about to begin our valedictory session. Now, I would like to request our participant to give their valuable feedback. Please unmute yourself and say something about our webinar. How did you find this webinar? Mm, how did it help you all, this webinar? So I am taking randomly uh, for your valuable feedback. So I would like to request Sir Adhiraj Dotto to say something about this webinar. Sir, please unmute yourself
Salma,
and say something.
thank you, Salma. It was a good session for the last days. I actually could not attend the full session yesterday, but about Lakhotia sirs and Sujoy's seminar, I have attended almost the full sessions, and it was nice. It was nice. I know both of them for the past few years, and I have nothing to say for Lakhotia sir. He is a legend, and I wish Sujoy every success in his endeavor that he is
Thank
going.
you. Thank you.
And thank you, KC College, for arranging such a good seminar.
Thank you, sir. Thank you, sir. Your valuable feedback. Now I'd like to request Shutopa Gupta. Ma'am, please unmute yourself and say something about this webinar.
Thank you. I am Dr. Sutapa Gupta from Dinobandhu Andrews College. I received this invitation via WhatsApp from one of my students. I attended all the 3 sessions. It was very well organized, and all the speakers are veterans in their fields, and they have given a very lucid presentation. So I think it will help the students and the faculty members at the same time.
Okay, ma'am. It's very nice of you. Now, I would like to request Dr. Saurabh Sarkar to say something about this session.
Thank you, Salma. I have attended all the sessions. I am from Gushkara Mahavidyalaya. I have attended all the 3 sessions, and the 3 sessions are wonderful. And the journey of 3 sessions, we have a enormous knowledge. Thank you very much.
Thank you, Saurabh. Now, I would like to request Abdul Sadat to say something. Sir, please unmute yourself and say something about the webinar.
Yeah, good morning to everyone. This is Dr. Abdul Shadat from Raiganj University. I had attended all the
Okay, sir.
3 sessions, and all these speakers are they are very eminent person in their field, and they enlightened us with their very eminent and very high impactful works, and it will be obviously help all of us, especially those teachers who we are in the university. They will be very helpful with this their works to polish their our own all the research works. I have a little bit of suggestion or a request. If you arrange some webinar where all the participant or we most of them can present any talk, that will be very helpful to promote our cash promotion. Thank you.
Okay, we'll try in our next webinar. Now I would like to take some students. Aditi Chatterjee, please say something about this webinar.
Yes, ma'am. I have attended all the sessions and The speakers were too good and they described in more lively pattern. We get in much more knowledge, though we have still yet to learn. But I enjoyed a lot, especially this lecture over Down syndrome. Ma'am, this helped me a lot. Thanks to the speaker and thanks to sir. Please conduct such webinars more and more in numbers. Thank you.
Thank you, Aditi. So, I now the feedback session is closed now. Now, I would like to request Dr. Pallab Jyoti Paul, Coordinator of IQAC, Assistant Professor, Department of Mathematics, Krishna Chandra College, to say few words.
Thank you, Salma. Thank you, Department of Zoology. Thank you, Joita. Thank you, Purnupama, other teachers, for organizing such an interesting 3-day international webinar. Thanks to our principal sir also. Thanks to Dr. Shamul Josh, our DBT coordinator. And thanks to Dr. Gorima Gupta, Scientist E, Department of Biotechnology, for offering our 4 departments, Physics, Chemistry, Mathematics, and Zoology, the prestigious DBT Fund under the standarding component of DBT Star College scheme. On behalf of IQAC of our Krishna Chandra College, I want to extend a warm welcome to the participants, because without the participants, this type of effort is meaningless. So my heartfelt thanks to them. We are delighted that all the participants have enjoyed a lot. I have understand from their feedback. We are privileged that there are so many resource persons, eminent resource persons, including Dr. Sujay Ghosh, present here today. We are privileged for their lectures. I also would like to convey my sincere gratitude for their exceptionally great deliberation. All the sessions I have seen are very good learning sessions. And thank you, Dr. Ghosh, for offering your time, your knowledge, your experience with us. We hope to see you once again in future. The session will end in few minutes today. I hope that there must have some major takeaways for the participants from this webinar, which will definitely help the teaching learning process and the research. Thank you. Thank you, Department of Zoology, for giving me the opportunity for presenting a concluding note. Thank you. Thank you, Salma.
Salmadi, please unmute yourself.
Okay, sorry. Now I request Dr. Shamul Kumar Josh, Assistant Professor, Department of Chemistry, Krishna Chandra College, and Coordinator of DBT Star College Scheme, to say something about the webinar. Sir, please.
Good morning, ladies and gentlemen. Is audible?
Yes, sir, you are audible.
So, good morning, ladies and gentlemen. It is a great honor for me to have this opportunity in this concluding note as DBT coordinator in this regard of international webinar on modern approaches and techniques for advancements of molecular biology, organized by the Department of Zoology, Krishna Chandra College, under the strengthening component of DBT Star College scheme. During the last 3 days session, it made us realize that science is becoming more developed for human being and nature. So that's why the students have really enjoyed this nice lecture by this honorable speaker. Thank you very much, Dr. Khatun, for giving me this opportunity. I would like to express my sincere gratitude to Mr. Naresh Chandra Bauri, President Governing Body of our college, Dr. Gautam Chatterjee, Principal of our college, and Dr. Pallab Jyoti Pal, IQC Coordinator, Assistant Professor in the Department of Mathematics, all the members associated directly or indirectly in this international webinar. In this context, I, coordinator DVT Star College scheme, am really proud to say that our 4 science departments such as chemistry, physics, mathematics, and zoology got DVT Star College scheme from Government of India, Ministry of Science and Technology, Department of Biotechnology, New Delhi. Thanks, many thanks to Dr. Garima Gupta Madam for giving us this opportunity to make various activities under this scheme. Again, I would like to acknowledge the effort made by the convener as well as the departmental coordinator, DVT Star College scheme, Dr. Joyita Mukherjee, assistant professor in zoology, the co-convener, Dr. Salma Khatun, meeting coordinator Purnapama Ghosh of the same department, and whole organizing committee for their enthusiasm and endless effort to make this webinar possible. I wish to express my gratitude to Chief Guest Dr. Shobhan Rai, ex-officio senior scientist, Government of West Bengal, Department of Science and Biotechnology, honorable speakers, Professor Subhash Chandra Lakotia from Banaras Hindu University, Dr. Shumedha Roy from Belgium, and Dr. Sanjay Rai from Calcutta University for being with us and giving their valuable time for their busy schedule. I convey my best regard to you on behalf of the team of DVT Star College scheme. I wish to have your physical presence one day to our college after escaping of the pandemic situation. Thanks to all the active and enthusiastic students and faculty members of our college as well as from the other educational institution and other institution for your participation in this webinar. Without you, program cannot be possible. I convey my sincere welcome for your kind participation. All of you have been given us this opportunity to meet all of you in this platform. Thank you all very much once again. Thank you.
Thank you, sir. Thank you for your nice words, and thank you for your support for the success of our webinar. Now, I request convener of the international webinar, Dr. Joyita Mukherjee, Assistant Professor, Department of Zoology, to deliver vote of thanks. Joyita, please.
Before I start, I would request Dr. Ghosh if he would like to say a few words.
Yeah. I just want to convey my thanks and my indebtedness to the organizer, particularly the Joyita, who contacted me to deliver this lecture. So I like to interact with the students because they are the future. So this is our responsibility to ignite their mind with some new information, so they could bear the torch after us. So this is very enjoyable moment, particularly the college, those who are at the outside of the metropolitan area is most important to me, because lots of brilliant minds lay there without getting much light. So I prefer to talk in those areas. I spent my 7 valuable years in the rural area and I found very brilliant students there, but they are not getting opportunity to come upwards and come to the city. But so your initiative to broaden their horizons with this webinar is really appreciable. I do appreciate all of you and congratulate you. Hope near future you will organize some physical mode of conference so that students will get opportunity to interact with the resource person in person. So thank you for all. Thank you for your organizing endeavor. Thank you.
Thank you, Dr. Ghosh. Thank you for your nice words. And we would like to organize such webinars in future. Thank you for your nice interactions with every participant. And I would like to request you, if the time, after this new normal condition, so when the college universities will be open again, so maybe if you permit us to take few of our students with to visit your lab?
Sure. My lab is open to all, and we run our lab with public fund, public money. So it's our responsibility to open up our door for the students, those who are interested, and I think they would be inspired to pursue research after visiting such lab. Not only my lab, but others' lab as well. So sure, I'm always ready to cooperate.
That will be great. Thank you. Thank you for your support. So now this is a time for the formal Thanksgiving. So very good morning to to our most valued honorable chief guest, patron and vice patron, worthy resource persons and all the participants. It is my utmost pleasure to acknowledge the contributions of those who worked really hard to make this webinar take place. I, Dr. Joita Mukherjee, on behalf of Krishna Chandra College and the entire fraternity of this college, first of all, extend my hearty vote of thanks to the Honorable Patron, Mr. Naresh Chandra Bauri, President of the Governing Body of Krishna Chandra College, and Dr. Gautam Chatterjee, Principal of Krishna Chandra College, for this their notable presence and wholehearted support to make this event successful. On behalf of the organizing committee, my most sincere thanks to all the distinguished speakers for their efforts towards making this webinar really brainstorming sessions. I must mention our deep sense of gratitude to Professor Subhash Chandra Lakhotia sir for his gracious presence and extremely knowledgeable talk on Drosophila genetics. I would like to express my sincere thanks to Dr. Sumedha Roy for giving an interesting talk on neurodegenerative disorder. I also express my heartfelt thanks to Dr. Sujoy Ghosh for his excellent lecture on Down syndrome. We are extremely gratified by the enlightening and well-informed talks by the honorable speakers. My sincere thanks to all the distinguished speakers for gracing the event and sharing their knowledge with the participants in spite of their extremely busy schedule. An event like this cannot happen overnight. It requires planning and bird's eye for the details. We have been fortunate enough to be backed by a team of very proactive and dedicated staff of our institution who are well versed in their jobs. I'm short of words for their involvement and their willingness to take on the completion of tasks beyond their comfort zones. A special mention to all of my colleagues of Department of Zoology, especially Dr. Salma Khatun and Mrs. Purnapama Ghosh. Their tireless efforts is a key factor that makes this event successful. Their enthusiasm, positive mind, hard work, and team spirit are the pillars for this event. With a deep sense of appreciation, we thank our coordinator of DBT Star College scheme in the Krishna Chandra College, Dr. Shamal Kumar Josh, for his persistent efforts and encouragement. I would like to thank all the faculty members of internal core committee of DBT Star College scheme of Krishna Chandra College. I do acknowledge Dr. Pallab Jyoti Pal, the internal quality assessment cell coordinator of internal quality assessment cell of our college, for his constant support and help and encouragement. I'm immensely thankful to Mr. Arindam Ghosh, Faculty of Department of English, Krishna Chandra College, for helping me immensely in technical issues to conduct such a meeting in online mode. I also extend thanks to all the teaching and non-teaching staff members of the college for their enormous cooperation to organize this event. I would like to thank our participants that made this event joyful with their inquisitive minds. Finally, I'd like to thank Dr. Gorima Gupta, Scientist E, Department of Biotechnology, Ministry of Science and Technology, Government of India for funding our college under the strengthening component of DBT Star College scheme to conduct such program. Thank you all. Thank you once again. So with this note, we conclude this international webinar. And thank you all. Thank you to Dr. Ghosh for being with us for such a long time in spite of your busy schedule.
Thank you.
Thank you and bye-bye. participants, please send the feedback link within 2 p.m. and follow the WhatsApp group for further details. And the e-certificate will be issued to those who have who will submit this feedback form, and it will be issued in next 2 weeks. Thank you.
Ma'am, there is no feedback form link in the description in the chat box actually. So how will you submit it?
Is it? I think it is there.
Oh, now it is there. Thank you, ma'am.
It was there, but
It was there, but there are a lot of questions there after that and
Yeah,
feedbacks.
yeah, ma'am. Thank you.
Okay, thank you.